Hi, my name is Justin Reese. I'm a research scientist at Berkeley Lab. So I'll tell you about KG Hub, which is our ecosystem for facilitating discovery of biological and biomedical knowledge using knowledge graphs. So a quick summary of what I'll talk about. I'll give you an intro to knowledge graphs if you're not familiar. I'll detail a couple of use cases, specifically our KG COVID-19 use case. Then I'll go into KG Hub, sort of our goals and design patterns. All of the tooling we have integrated. I'll briefly go through the 9 or so KG Hub projects that we have. Now I'll do a quick GraphML demo. GraphML is one of the use cases that we're most interested in applying to KGs. So we'll train a GraphML model in 9 lines of Python code. And then finally, I'll go through some of our ongoing work to try and leverage LLMs for constructing KGs. Here are the slides if you want to follow along. So what are knowledge graphs? Conceptually, this is very simple. Entities, people, places, genes, concepts, those are all represented as nodes on a graph. And then the relationships between these entities are represented by edges between those nodes. So this is a really flexible and easy way of adding new data, at least compared to a relational model. This gives us really rich representation of complex data, both entities and their relationships. And this is a really elegant way of integrating multimodal biological data. So for example, here is a paper from the Zitnik lab, where they've integrated a lot of biological data from a lot of different sources. So the green arrow represents data you might get from a clinical trial, drug treats disease. The red line would represent maybe epidemiological data. This exposure causes this disease. The blue line might represent data that you will get from RNA-seq. So this gene is expressed in this anatomical region. So this is a really elegant way of combining data from a lot of different domains in biology. So what can you use these for? Obviously, if you go to the trouble of making a knowledge graph, it stores your data and also, as I said, represents your data in a really elegant way. This also is a really convenient way of querying and browsing your data. So this is good for generating hypotheses about your data or just generally familiarizing yourself with the data. One of our favorite use cases, though, is graph machine learning. So graph machine learning on KGs was famously used to create social networks that I'm sure everybody's familiar with, Facebook, Twitter, and a lot of others. You can also use KGs to generate new biological knowledge, or what BOSC termed network medicine. We made some contributions in this area too. We have this GraphML package called GRAPE, which I'll go into later. We made a KG to integrate COVID-19 data called KG COVID-19. We've also done a lot of other work on GraphML algorithms and actually applying them to biological data. When the COVID-19 pandemic hit, we put together a COVID-19 knowledge graph that we call KG COVID-19. But this underscores a theme, I think, which is in biology, lack of data typically is not the problem. So before the pandemic even hit, we had data about SARS-CoV-1, that was a thing, MERS and other coronaviruses, and then SARS-CoV-2 hit, and people started generating just droves, massive amounts of data, drug data, viral host PPIs, sequencing data, Go annotations, and just an endlessly longer list than that. And of course, there's more SARS-CoV-2 data by the minute. But really the main challenge early on and even now is data integration. The data tends to be siloed, it tends to be trapped in text. It's behind separate APIs or in various file formats. And this problem doesn't just apply to COVID-19 or biology. This problem comes up in many other data science domains. And so we constructed KG COVID-19, at least to solve this problem for COVID-19 data. Here is a high-level view of what is in KG COVID-19. So we have about 60,000 drugs, 50,000 or so publications, all Go terms, all human diseases, all human phenotypes, human genes and human proteins, and of course SARS-CoV-2 and other coronavirus genes and proteins. So we released this COVID-19 knowledge graph as a community resource. It's currently being used in N3C. It was also used in the National Virtual Biotechnology Laboratory, which was a DOE effort to address the pandemic. You can read more about it in this publication here. We also applied GraphML on KG COVID-19 to see if we could find some drugs that might have some effect on SARS-CoV-2. That's what I'm showing you here. If you embed the nodes in KG COVID-19 into a simple experiment to see how close in embedding space the drugs are to SARS-CoV-2, you see this nice Gaussian distribution, and then you see this shoulder off to the right, which likely represents drugs that probably have some direct or indirect relationship with COVID-19. So we did a deep dive on a few of these, and it turns out others were looking at these drugs also, and so we collaborated with them to sort of corroborate what the GraphML was telling us. You can read more about our work on metformin drug repurposing and the effect of NSAIDs on SARS-CoV-2 too. So what's the problem? Well, the problem with KGs is the ecosystem we've created as scientists is not very fair. The KGs themselves are not typically very findable. They're strewn across the internet, or they're only mentioned in passing in papers, and it's actually pretty hard to find a URL to download them. Or worse yet, they're not in the internet at all. If you can find them, they're typically not very interoperable. There's not a common data model really that everybody agrees with for representing the data, which admittedly is very hard. They're typically not very reusable. So how specifically a KG was built is pretty unclear typically. There's a lack of provenance. Where did the data come from? How was it transformed? And it's also very difficult typically to add or remove components. So if you have a KG you're interested in, you want to layer your data on top of it, that's typically kind of hard. And there's also no standard operating procedure for building KGs and no standard way of doing QC. So the result of this is a lot of duplication of effort and a lot of frustration and just generally slower research. And so our goal in broad strokes in creating KG Hub was to address that. So we wanted to create a knowledge graph ecosystem with these properties. So sort of a federated KG of a KG ecosystem based on a set of useful patterns that we think is a good way of doing this. So if you're familiar with Obo Foundry, this is a similar idea for ontologies. So they're not necessarily completely aligned, but they're a federation of ontologies that were created with a common set of principles in mind. So the idea here is Obo Foundry for KGs, if you like. We want to preserve provenance, maximize reproducibility and fairness. And so we also want to allow remixing of subgraphs. So it happens a lot that you might be interested in incorporating string PPIs into a knowledge graph. And so KG Hub allows you to go find a pre-transformed version of string and download it. We provide tooling for less painful downloads, for less painful transforms from raw data into graph format, ID normalization. We align to the BioLink data model. This is a high-level biological data model that makes it a lot easier to mix and match knowledge graphs between projects. If people align to the BioLink data model, then we know where their genes are, where our genes are, and we can more easily align them. We're very fond of incorporating ontologies into knowledge graphs, so we made it easy to incorporate any of 204 Obo ontologies. Made it easy to do QC to sort of quickly assess the contents of KGs and their topological features. It's very easy to browse, all the data and artifacts for a project online. We've also gone to great pains to make it easy, fast, reproducible GraphML possible. So how does all this work? So typically, when you're building a KG, you have this upstream data that often is in tabular form. So here's a list of patients and their conditions. And then, as I said, we're very fond of incorporating ontologies into knowledge graphs. So those are pre-transformed into something we call KG-OBO. Here's a list of diseases and genes that are involved with those diseases and drugs that affect particular diseases. So we apply ETL tooling, and that breaks down in 3 steps. We have a download software called KG Hub Downloader. This is a frequent point of failure in ETL, and so we've made this fault tolerant and sort of re-entrant, if you like. After the data is downloaded, we provide a tool called COSA, which allows for simple declarative transform of typically tabular data into GraphML, into graph format. Those graphs are combined into one graph using a tool called KGX. And so you can see down below, the data that started off as tabular is now in graph form. And so this provides, this sort of creates what you might call emergent knowledge. So it creates sort of relationships, for example, between patients and drugs that were not present in the original data sources. And this provides a really good substrate for a lot of different use cases, particularly GraphML. Another one of our goals was to make it easier to sort of remix KGs. So it's been typically pretty difficult to find a subgraph that somebody else has transformed of, say, like I mentioned, string. If you want to incorporate that into your KG, you would typically do that yourself. This provides a way of going and getting somebody else's transform of a string and incorporating it into your KG. All of this, the ETL process for KG Hub is YAML driven. So, for example, how to download the data and where is specified in a download.yaml file. This is fed to KG Hub Downloader, which downloads the data. The transform that happens, and makes subgraphs for each of the upstream data sources. These can be recombined according to a merge.yaml file. So you can mix and match all of the components, the subgraphs, in a way that builds you a KG that's exactly what you want. We take care by virtue of COSA of ID normalization, and as I said before, we align with the bilink data model that's provided for in COSA automatically. And so all the data is aligned to a data model that makes it easier to mix and match with other projects that are also using the bilink data model. Here are the current KG Hub projects, 9 of them, I think. KG COVID-19, I mentioned before. KG Microbe integrates microbe data. KG IDG is integration of data and of drug and related data for a project that we participated in called Illuminating the Druggable Genome for drug discovery. KG Monarch is a KG representation of the Monarch graph. We work closely with a lot of members of the Monarch Initiative and developed a lot of the tooling I just described with that team, of which we are a part. EcoKG is integration of plant genotype and phenotype data. As I said before, KG-OBO is sort of a pre-transformed of all OBO ontologies, 204 of them. KG-Phenio is a subset of KG-OBO ontologies that we frequently use. So we pre-transform those into a sort of a modular set of KG ontologies that we can use for KGs. These follow common design patterns, as I said. The ETL process I just described. We follow a property graph model that's represented in a format we call KGX TSV, which is a simple TSV format. COSA gives us simple declarative transforms, and as I said, these are easily remixable. We provide stable URLs for all builds. So typically in a monthly cadence, each of these is rebuilt and put somewhere with all of the artifacts, all the subgraphs, all the provenance data, in stable URL format. And we provide a lot of downstream tooling too that can be applied to these. So here is a schematic representation of all this and how all of these parts interact. We host the ETL code in GitHub, so the software developers who's interested in making a new project would create a new project under the KG Hub organization in GitHub. Whenever that code changes or a month passes, typically we would do another build using a CI tool called Jenkins. Optionally, we can use KGX to output data directly into a Neo4j or BlazeGraph database, or freeze this off as files. As I said, one of the main use cases we're interested in is GraphML, and so we helped develop this package called Grape, which I'll go into in a minute. If you have a GraphML task that you're interested in doing on every monthly build, then you can specify that using a tool called Neat. That is also YAML driven, so every time a new build appears, you can redo in a reproducible way the GraphML task you're interested in. Also, by virtue of aligning with Grape, we have API access to all KG Hub graphs. So for example, if you're interested in the latest version of KG COVID-19 and you're in Python, you can just say from grape.datasets.kghub import KG COVID-19. All these builds go directly into the cloud after we build them, S3, Amazon. The homepage points to these artifacts. You can also browse all of the files in your browser. This provides full provenance for all the data that was used to generate the KG. So all the code and all the data that you can use to rebuild the KG lives on there. We have summary statistics, graph artifacts, and all the subgraphs too, if you're interested in just one part of the previous build. We provide tooling to display the contents of the KG. So, here's a KG, what BioLink node types and edge types exist in there, and what exactly is ingested from each source. As I said, one of our main interests is GraphML on these KGs after we build them. So, as I said before, we provide programmatic access to KG Hub KGs via Grape, and we think that the combination of these two is a really powerful tool for reproducible GraphML. So here, for example, is an entire reproducible pipeline to download a version build of KG IDG from July 2021. This is frozen off; it will never change. Make connected holdouts for a test train split, generate node embeddings, train a perceptron edge prediction model, all in 9 lines of code, and on my laptop anyway, less than 20 minutes of runtime. So this works now. You can download this code. It should work the same on your laptop. It should work a year from now and 5 years from now. Another future direction we're currently pursuing is using large language models like GPT-4 to build KGs directly. So the way this typically works now is scientists do experiments, they write up their knowledge in scientific literature, that eventually, in an incomplete way and kind of a slower way, percolates its way into databases. The databases typically are what we download to feed into ETL tooling that I described before. The idea here, though, is to use large language models to directly import knowledge from scientific literature, and so sort of short circuit this circuit right here and go directly from scientific literature into graph data. This is possible because of a tool called OntoGPT, which Harry Caulfield will be talking about, poster A087. You can also read more about it in this preprint. So the idea here is, as I said, to leverage LLMs to ingest knowledge directly from scientific literature. And so we're investigating this to sort of build in an automatic way bespoke KGs for a particular disease domain. So if you're interested in inflammatory bowel disease, you can automatically query PubMed, identify the papers that might be of interest, and directly import their knowledge and kind of layer it on an existing KG with things like biomedical ontologies and other data from databases. Some quick acknowledgements. Harry Caulfield was instrumental kind of down in the trenches building a lot of the tooling that I described. Chris Mungle also was involved and the entire BBOP crew from Berkeley Lab. We work very closely with Peter Robinson's lab at Jackson Lab. We also worked very closely with Giorgio Valentini at University of Milan and Elena Casaraghi. Luca and Tommy did most of the development on GRAPE, which I described. We worked very closely with Melissa Handel at CU Anschutz, and Tiffany Callahan is the developer of Phenolator, which you may have heard of. And so we sort of in parallel developed a lot of these design patterns and tooling with her. Thanks for listening.